Okay, so may I start?
Please go ahead.
Okay, yeah, thank you everyone for your time today. And I'm very honored to be the first presenter of this summer computational neuroscience speaker series. And today, as a first talk, today I'd like to speak about these, mainly like these two things, the estimation of neural interactions and the detection of cell assemblies. I will also touch a little bit about the how the data analysis and the modeling work together. Okay, so that's the plan for today. Okay. Before we get started, because this is a very first talk, I'm thinking to discuss about the idea about the computational neuroscience. So I put like 5 images of the like important computational neuroscience textbooks. They cover from the data analysis, and they also can talk about the data analysis and the modeling together, more purely modeling. That's this book. And this is a relatively old textbook, but this is a great introduction to the theory of neural computation. So as you can see, when we say computational neuroscience, that will cover from like a data analysis to the modeling and to the theory. So we can probably summarize that the computational neuroscience is the field of study that investigates the brain function using mathematical tools and theories. Okay, and I have also prepared another slide talking about the difference between the machine learning and the computational neuroscience, because probably many of you have heard of this machine learning. That's a really big thing recently. And what is the relationship between these two things? So I'd like to say that machine learning is the field of study that develops the systems that can automatically learn and improve from experience. Okay. So again, I put like 4 kind of important textbooks about the machine learnings. So the Bishop book, and also the Kevin Murphy's book. Deep learning, I also included deep learning and reinforcement learning. They are the subfield of the machine learning. And actually, in this deep learning textbook, I found a very good introduction about the relationship between machine learning and computational neuroscience. So they say, actually, their case, they say deep learning is primarily concerned with how to build computer systems that can solve tasks requiring intelligence. So that's the deep learning. While computational neuroscience is primarily concerned with building models of how the brain works. So we can probably say that the machine learning, deep learning, reinforcement learning, they are the fields in the engineering which try to develop the system which can behave like humans do. On the other hand, the computational neuroscience, we primarily focus on to understand how the brain works. So that's a science. So that's a difference, engineering and the science. Okay, so that's a kind of brief introduction about the computational neuroscience and machine learning. So these are the 3 topics for today's talk. I'm going to start talking about the estimation of the neural interactions. We're going to discuss why this is important and how it can be performed. Next, I will move to the detection of cell assemblies. I'm going to talk about what are the cell assemblies and how they can be detected. And finally, I'd like to talk about, so these 2 things are more like data analysis. So finally, I'd like to talk about how the data analysis and modeling can work together to deepen our understanding for the neural information processing. So as I said initially, I'm going to stop at the end of each topic. And then if you have any questions, please feel free to ask me. Okay. Okay. Oh, okay. So let's get started about this first topic. So this slide is the basic neuroscience introduction for the people who are not very familiar with. So Here I put the image, like a staining the neural tissues, and you can see some, black objects, these kind of things. So they are the neurons, and you can see also the, neuron has some, complex morphology, and so that's the image of the staining. And this was the kind of famous drawing done by the Kahar about more than 100 years ago. So he was drawing how the neuron looks like. Okay. So from these kind of studies, what we now know is that the nervous system is made up of discrete cells, and we call them neurons. And we also know that neurons are connected by synaptic junctions. So in this picture, it looks like maybe the nervous system was continuous, but it was not. So we see the distinct units called the neurons, and they are connected by the synaptic junctions. Okay, so that's the basic neuroscience part. So now I want to talk about neuroplasticity. What is the key feature of this nervous system is that the nervous system is able to change their structure, like physically or chemical properties. And this is indeed the hallmark of the nervous system functioning. And we also know that this neuroplasticity is required for learning and memory. Okay. So how this happens, we have the very good insights coming from the Canadian psychologist, Donald Hebb. In his famous book, published in 1949, he proposed this concept. The synaptic connections are modified by coactivity of the connected neurons. So the depending on how the two neurons are co-activated, the relationship between them can be modified. So in short, we can say that like cells that fire together, wire together. So this kind of one way of saying. Of course, this saying says when the two neurons fires, the connection gets kind of strengthened. There is a cases that the connection gets weakened, but the key idea was summarized here. The coactivity of the neurons are important to the change of the connection happens. Okay, so this is the key feature of the nervous system. Okay, let me go to the next one. So therefore, now we see that when we talk about the brain functions, we really need to pay attention to the connectivity of the brains. And when we talk about the connectivity, we should probably be aware of the three kinds of connectivities we can talk about. The first one is called structural connectivity. This means the neurons are physically connected. So these are physical connections, which is kind of depicted here. So when we talk about different areas of the brain, we can find the area they are physically connected by the synapses. So this is the structural connectivities. They are typically measured by the putting the tracers to the fibers, or the non-invasive diffusion tensor imaging. So those are the techniques to investigate structural connectivity. Another type we say functional connectivity. This is the statistical actually property, but we don't really talk about the directionalities. So it's a non-directional statistical interaction. And so that's the middle one here. Okay, so we can talk about how the areas are co-activated, like simultaneously. But we don't talk about which area drive the other the area. Okay, so that's a non-directional. And the measures for this functional connectivity are often like correlations, coherence, or like a mutual information. So you can see that often the functional connectivity is summarized in this correlation matrix. And it's important to notice that they are the symmetric matrix across these diagonal lines. Okay. The third one is called effective connectivity. This is the one which we talk about the directions. So the directional effects, that's kind of summarized here. And you can see the difference between this and this Now we see the arrow here, and Y is maybe thicker than the other one. And it's a bit challenging to see here, but this is indeed asymmetric matrix because there is a directionality. Okay? And the measures used for this type of connection is like a Granger causality or like a transfer entropies or like a dynamic causal modeling. So we can say that this is the physically connected patterns. These two are more like a statistical properties, non-directional ones and directional ones. Today, I will mainly focus on these functional connections because this is quite often used in many different experiments. And this is also I'm primarily working on right now. Okay, so we're gonna focus on this functional connectivity today. Okay, so I mentioned that to estimate the functional connectivity, we often use correlation measures. And one of the measures which is often used is correlation coefficient, which is defined something like this. Here, the eta is the firing rate of the neurons. So eta 1 is the firing rate of neuron 1. Eta 2 is the firing rate of neuron 2. Eta 1, 2 is the joint firing rate of two neurons. So by observing these three quantities, we can compute the correlation coefficient. Because this is very easy to use and it's very simple, it is often used as a measure for the functional connectivity. So this is great. However, there is one issue which I pointed out here. The correlation coefficient is in fact dependent on neurons firing rates. So let me describe what this means using this figure. This was a study I performed with my collaborator, Jean-Marc Fellows at the University of Arizona. He actually performed the biophysical network simulations using the neuron simulator. So the activity of each neuron looks something like this. So it has the action potentials, looks like a realistic activities. And he has connected 5 neurons in this case. And you can see the blue lines. So these are the connections. And he actually did 2 different simulations. One, he modified the connection weights. So you can see the thickness of the blue line increases. Okay. The other case, what he did was that he maintained the connection weights. So you see the thickness does not change, but he was increasing the background inputs from, let's say, from other neurons. That was depicted by this red arrow. So you can see the red arrow gets thicker. Okay. So we say in this case, because connection weight is modified, this is more like learning. This case, connection weight is not modified, so we say there's no learning takes place. So these are the 2 simulations. Okay. So now we are plotting how the mean firing rates of these these neurons changes over 3 distinct epochs. He was increasing the connection weights in a discrete way, so we can actually see the firing rates increases in a stepwise way, corresponding to the increase of the connection weights. Similarly, when he increased the background inputs in a discrete manner, we can see the very much the same change of the firing rates. So this indicates that by looking at the firing rates, we cannot distinguish what is actually going on, either the learning or just the background inputs is increased. They both shows up in the firing rates. Okay. Now let's look at how the correlation coefficient behaves under these 2 simulations. Under the learning scenario, where we increase the connection weights, yes, we see the discrete increase of the correlation coefficient. This is good because this should supposed to detect the functional connectivity change, which is increasing. The issue is here. Here, we also see the correlation coefficients is almost like a discrete way increasing. However, the connection weight is maintained the same. So therefore, this is showing that this is depicting the increase of the background inputs, which is reflecting to the firing rates. Okay. So this was the example that I'd like to say that correlation coefficient is also dependent on the neurons firing rates, and not only to the connection weights. Therefore, by using the correlation coefficients, we cannot really tell what is going on here. Okay. So as one of the studies, we are trying to develop the new functional connectivity measures based on the study called information geometry. I had an opportunity to work with the Dr. Amari, who is one of the founders of this information geometry study. So in one word, we can say that information geometry is the field of study that applies differential geometry to the probability theory and statistics. And it appears quite different from the neuroscience, but it turns out that this is quite useful to investigate the functional connectivity. So let me describe how we can derive the new measure for the pairwise interaction. Let's assume that we have two neurons. We are focusing on two neurons. Neuron number 1 fires like 3 spikes here. Neuron number 2 spikes again, 3 spikes here. Our first approach is that we're gonna represent these firing patterns by the binary random variables. To do this, we can bin the activity into the small bins like this. So this activity from neuron 1 is converted to these binary numbers. So there's no activity here, so that's 0. No activity here, 0. We see spikes in this bin, so we say 1. No spike, 0, something like this. So you can see this spiking activity is converted to these binary numbers. We do the same thing for the neuron number 2, spike activity here. They convert to 1, 1, 1 here. Everything else is at 0. Now we see there are 4 mutually exclusive events between these two neurons. We see 0, 0 events. We see 0, 1 events. We see 1, 0 events. We also see 1, 1 events. So these are the 4 different events we can expect. And by observing the data and using the maximum likelihood ideas, we can estimate the probability of these different events by this way. So we say the probability of the 0, 0 events can be estimated, the number of these events divided by the total number of the bins, divided by the total number of bins. Similarly, the event for the 0, 1 can be estimated by the total number of the 0, 1 events divided by the total number of bins, same way. So from this observation, we can construct P00 to P11, okay? Now let's move on how we can construct the information geometric measures. By having the binary representations, we know that probability distributions can be exactly expanded using the log linear model, which is described here. So this is called the log linear model. We have the log and it's linear to the coefficient. And now in the previous slide, we can estimate this P00 to P11. So we can plug this into this equation. So let's say, for example, when we talk about P00, that means both neurons are not active. We put 0 and 0. So 0 here, this term goes away. 0 here, this term goes away. 0 here, this term goes away. So only this term remains. So we have the equation like this. Log of P00 is equal to minus 5. Same way we can do 0, 1. We get this relationship. We do the 1, 0, this relationship. Finally, we do 1, 1. So all these terms remain, survives. So we have theta 1, theta 2, theta 1, 2, minus 5. We can solve these equations for theta 1, theta 2, theta 3. And finally, we come to these expressions. Theta 1 is given something like this. Theta 2 is given something like this. Theta 1, 2 is given something like this. Now, these are called information geometric measures. And especially when we focus on this theta 1, 2, which is the coefficient of the correlated activity. This is the new measure we are proposing to replace the correlation coefficients. So correlation coefficient was derived, given something like this. Now we are saying that the two neuron interactions can be estimated by this equation. Okay? And as you can see, everything here is that the probability of the 4 events, which can be estimated from the observation, right? So now let's analyze our simulation data using this information geometric measures. That is summarized in this lower panel. Okay. So first, let's focus on this blue dotted lines. This is corresponding to the second order information geometry. Let me see, that's this one. We try to replace the correlation coefficients. Okay. So now when we look at this first simulation where the connection weight is increased in a discrete manner, we see the discrete increase of the 2 neuron pairwise information geometry. This is good. This is expected. But importantly, please look at this portion. When we analyze this scenario where connection weight is maintained the same, we see the flat change, not really change in this pairwise information geometry. So when we look at using the pairwise information geometry, we can actually see this is the case where the learning takes place, but this is the case where no learning takes place. We have the additional information coming from the first order info geo, theta 1 or theta 2. Now we see this is pretty much the flat. Here we see the discrete kind of increase. This is corresponding to the background inputs. In this simulation, background inputs, red arrow, was maintained the same, so they stay the same. But here we increase the background inputs in a discrete manner that was depicted here. So using the information geometry, we can separate out the change in the connection weights or the change in the background inputs. So this is more informative than using the correlation coefficient. Okay, I believe this is pretty much end of the first section. So I have shown that the so this is the summary so far. So we talked about why we want to focus on the neural interactions. This is because the neuroplasticity is a key feature of the nervous systems, and the neural interactions. By looking at the neural interactions, we can get the crucial information about the neuroplasticity. Okay, and we are focusing on functional connectivity today, and there are many measures for that. But I've shown as one of our study that the purely pairwise interactions can be estimated using the information geometric measure. Okay, I think I will stop here, and I'd like to answer if there's any questions.
Hi, Masami.
Yes.
So would you please explain why you said that the probability distribution had to be linear, log linear?
Okay, let me go back. This portion.
Yeah.
Okay, so actually, this not has to be actually the, so when you take a log out, so this is going to be exponential and this is exponent. Yeah. And yeah, so this is actually the exponent family distributions. And actually, what I tried to say was that if we construct the probabilities, discrete probabilities like this manner, this belongs to one distribution of this exponential family. So the exponential family is actually quite broad, including the like normal distributions, Poisson distributions, binomial distributions, But as one of the distributions included in the exponential family, this discrete one is actually included. So that's how we can expand using this property.
Yeah.
Okay.
And does it have to do anything with the maximum probability principle somehow that you came up?
Sorry, your question is that it has to do that something has to do with this maximum likelihood estimate. Well, this part is actually different to my understanding. So, so the P00 to like a P11, yeah. So as long as this can be estimated reliably from the experimental data, that's the important part. And for this example, we used the maximum likelihood estimate to estimate the P11. But if there's any other better way to do that, that's fine. But as long as we have the P, discrete probabilities like P00 to P11, this portion is not affected by how we estimate.
I understand. Thank you.
Okay. Okay, Jennifer, should I move forward or any other questions?
I do not see any other questions in the chat. Is there any other questions from anyone else? All right, I think you can proceed.
All right, thank you. Okay, so let me move on to the second topic. The second topic is about the cell assemblies. So I want to briefly talk about what are the cell assemblies, and I want to discuss about how to detect those. So this is another topic of the data analysis in the computational neuroscience. So the cell assemblies, again, this important concept came from this the Donald Hebb, his famous book, 1949. So in his textbook, so this was the figure from his book. He talked about this like a diagram where the arrow represents simple assembly, meaning that in this case, like a neuron 1, 2 fires, here neuron 1, 3 fires, neuron 5, 9 fires. So he's kind of picturing that there's a sequence of the simple cell assemblies, kind of get activated. So we can probably summarize the cell assembly is the idea in which, so this represents a group of neurons that is being activated repeatedly during the certain mental or perceptual process. Okay, so why we want to focus on this? So we are not kind of thinking that, so there's a, let's say, maybe stage, like a theater stage set up by the brain connectivities, brain connections. Then on this like a theater stage, there are many actors, playing. Those are the cell assemblies. Okay? So we want to also focus on those actors, not only to the theater stages. Okay? So I want to first give you how this cell assembly looks like in our experiments. This first example came from the study performed by the Dr. Mike Eckhart. So what he did was that he trained animal for the so-called rat skilled reaching task. In this task, rat is trying to reach to the small food. You can see the small white stuff here. So this is located outside of the cage, and there's a small opening window he can reach out. So animal tries to reach this food, and he can extend his paw, grasp, and eat. And this actually requires some training. So this is called a skilled reaching task. So Mike was training the animal for this skilled reaching task, and simultaneously he was recording the neurons from the primary motor cortex. So these are the trial 14 to 16, and this comes to this bottom panel. So these are the 6 trial examples when animal did this movement, reaching task. So here we see about 75 neurons recorded simultaneously, and the like orange-ish color. These are the I believe, and this dashed blue line is indicating when animal grasp the food. So we can see some neuron get activated before the reaching activity, during around the grasp performance, then after that. So we can kind of see the nice sequence of the neurons activity here to here, again here to here, here to here, yeah. So by averaging these individual trials, we can get the average neural activity, something like this. So this is the average picture, and now we can more clearly see some neurons are active before the grasp motion, some neurons are active during the grasp, other neurons are active after that. And we often say this is the templates. Okay? So this, I'm showing that this is one example from the cell assemblies. Okay? So once we can construct the cell assemblies, using like this template approach. One of the methods often used to detect cell assembly is called template matching. So the template matching is basically moving that templates over the actual recording patterns and try to detect whenever the similar pattern shows up to the templates, we say that neural activity is a candidate of the cell assembly. Okay, so the template matching is to investigate the repeated activation of cell assemblies. So this is another our classic study we published quite many years ago. So in this study, animal was running on this circular arena following the certain sequence. Okay, and we started recording from the sleep one. We trained animal for this task. Animal sleep again. We bring back the animal to the arena again. That's the second maze session, and he sleeps again. And during all these epochs, we keep recording the neurons from the prefrontal cortex. And as I described before, we can actually construct the templates. In this case, I actually selected templates, which is corresponding to the one path. Let's say that is running from here to here. This is my important behavior epochs. So I selected the neural activity when animal behaves running to here. That's corresponding to the neural activity, something like this. So we have the simultaneously recorded neurons over a certain time. So we can again see the neural activity firing initially, that's corresponding to around here, to the neurons around here, which is active at the end of the behavior. Okay. So we can run the template match using these templates. And interestingly, we can actually find that there is a similar pattern shows up again during the post-task sleep. So meaning that after task training happens, like, post-task sleep here, or the post-task sleep here, we can see the highly similar patterns to the templates. This is the one example. And this is called like a memory replay or memory reactivation. Okay, so far, I hope this is reasonably good. But there's actually one issue of this template match analysis that is described here. The template match relies on the reference events. For example, like a behavior or the stimulus, meaning that I said I selected the templates based on the certain behavior. But in this experiment, there may be many other cell assemblies which I didn't select. But in the template match analysis, it's probably missing very many those cell assemblies. So it is important to develop the another cell assembly detection methods which does not rely on the reference events. And there are actually, this is kind of getting hot topic. There are several approaches. I'd like to describe one approach. I have been working with my collaborators in Japan. Actually, they are really the main contributors. I typically provided our data sets. My contribution is a bit small, but I'd like to describe this approach. So this is the one approach we say the cell assembly detection without reference events. And we are using the idea so-called edit similarities. The edit similarity is the concept from the computer science. And this is actually try to find how many manipulations is necessary to match one sequence to another. So in this example, we have two sequences, ATCG-TAC. Another one was ATGT-TAT. So we can so the idea was that how many manipulations is necessary to match these two sequences. Okay. And we can see this like a matrix in the center. So we see, let's say, initial letter is same A, the second letter is the same T. So we move actually on the diagonal, and we are counting how many matches happens. But third one is a G, here is a C, so there's not much. So therefore, to make this sequence align, we have to kind of insert new letters or delete some letters. Those are corresponding to the horizontal movement or the vertical movements. Here we don't see the increase of the numbers. Only when we see the diagonal movements, we see the increase of numbers, which is corresponding to the match of the same letters. So idea here is that by doing this kind of manipulations, in the end, we see the 5 here. that's corresponding to, there are 5 matches of these 2 sequences. Okay. So briefly, that's the idea of the edit similarity, and we apply this idea to match 2 similar cell assemblies. This is the results, what we got. So this part of the figure is corresponding to this portion. So we have sleep 1, maze, sleep 2, another maze, sleep 3. So the cluster, we detected several clusters, and we see all those clusters are active during the maze sessions. But we also see those clusters tend to reactivate during the most strongly during the last sleep session, a little bit during the middle sleep sessions, but not much during the first sleep session. We show 3 representative clusters, and they are the corresponding behaviors. Now we see these behaviors quite different from what I selected here. I selected behavior running here to here, that's my template. But now here the repeated sequence, cell assemblies, corresponding to completely different behaviors. So these are the examples that the repeated cell assemblies that was missing in my template match analysis. But now this was detected using the detection method without reference events. One more example I'd like to show you is that we were also interested in the detecting the how many cell assemblies possibly exist in the old and young rats. Our hypothesis is that so when animal is young, or we are young, we have better memories. When we get old, our memory function, unfortunately, worsens. So the one question was that, is it relating to the number of the available cell assemblies? So the hypothesis is that maybe the young animal has more cell assemblies available than the old animals. To ask these questions, we collaborated with the Dr. Szymanski and Carol Burns in Arizona. By the way, Dr. Szymanski, he graduated from the University of Lethbridge, and she became the neuroscientist. And so when she was a postdoc in Arizona, she performed the huge experiment. She ran the animal on this like a circular track. And when the animal is running on this track, at certain locations, the rats get stimulated on his eye. So this is called trace eye blink conditioning. So when we repeat this experiment, animals gradually run where he gets stimulated. So they tend to close the eye before they get stimulated. Get stimulated. So she ran 6 animals for this experiment, the 6 old animals for this experiment. And interestingly, one more thing I should say that for each day, she ran 2 recording sessions. So in the morning, she ran the experiment once. In the afternoon, she ran another experiment. Here, what you see is called remapping or non-remapping. In the hippocampus neurons, we know that the neurons are firing at the specific locations. So when you linearize this circular map, so this is a linear, a linearized map, you can see the neurons preferred place field along this map. So we can see very nicely the entire maze is covered by this group of neurons. This was constructed from the morning recordings. This was constructed from the afternoon recordings. We can see pretty much the same map, meaning that when the animal comes back to the same maze in the afternoon, they reproduce the same preference. And we say, probably, animal remembers where he was in the morning, and he records in the afternoon too. But in another recording session, so this was the map from the morning, but in the afternoon, when the animal comes back, it's completely disrupted. This is called remapping because animal may not remember where he was in the morning, so they may be generating the new map, possibly, recognizing, oh, this is the new place in the afternoon. Okay, so the remapping and non-remapping, those are some interesting activities. Okay, so we have analyzed these data sets using the different techniques, but without reference events. And this is our preliminary results, which we thought it's quite interesting. So here I'm plotting the dots, which is corresponding to the each day of recording. This is the number of the detected cell assemblies. This is the number of neurons recorded in each recording session. The point here is that we see the dots around here. So the majority of the dots around here, but these are the outliers, which represent there very many cell assemblies. And you can see these are mostly the blue dots. That means non-remapping days. Non-remapping days means that the animal, when animal comes back to the maze in the afternoon, they remember the experience. So those are the blue dots. Here are the same results, but colored in a different way using the young or old. Now you see the majority of the dots are the red, corresponding to the young animals. Only small portion is the old animal. So putting together, we can say that those days that we see very many cell assemblies, they are the non-remapping days, and they are mostly young animals. So it's kind of interesting that I said, so the young animal, they tend to have the better memories, and the non-remapping days, they appear to remember where he was. So showing the good memory performance. Those are the days we see very many cell assemblies. So this is possibly supporting the idea that number of the cell assemblies may be a good measure to discuss about the memory function. Okay, I'm going to stop here, but let me just quickly summarize. We have talked about the detection of the cell assemblies. Cell assemblies are the group of neurons that are being activated repeatedly. And we show that template matching has been widely used, but now it is important to develop the new techniques which can detect cell assemblies without using a reference event. Okay, so that's, I'd like to stop here.
Thank you so much for that. We actually do have a few questions. I think Javad had a question for you. Javad, are you ready with your question?
Yeah, I can ask that. Masami, if we detect cell assemblies using the techniques without using reference events, then how can we interpret the results? Let's say we found a couple cell assemblies, then how can we interpret the results?
So, sorry, how can we differentiate?
How can we interpret the results? Like what is the function of those cell assemblies that we detected without using the reference?
Okay, thank you. That's a very important question. So I think the point here is that for the detection of the cell assembly, we are proposing that we are not going to use the reference events. So it's more like a systematic detection of all the intrinsic cell assemblies. Once we detected all those cell assemblies, yes, now we want to correlate those cell assemblies to some other external events. So we can, maybe use the mutual information, to relate to them, but to interpret the meaning of the cell assemblies, I propose to use the external events.
Okay, thank you.
Yeah.
And there is another question from Ayush. In various experiments discussed in the second part of the talk, which part of the brain was selected and what techniques was used to record brain activity?
Thank you. Sorry, I forgot to mention about that. So I mainly talked about, so here, this experiment, so all the experiments we are using the multi-electrode recordings from the freely behaving animals. And so this was, so when we trained the animal for the skill reaching task, we recorded from the primary motor cortex. For this sequence task, we recorded neurons from the prefrontal cortex, primarily from the prelimbic, prelimbic areas and anterior cingulate areas. For this, this is actually the same recording data sets. And for this recording, they recorded from the CA1 of the hippocampus. So 2 experiments were from the cortex, primary motor cortex, prefrontal cortex. The 3rd experiment was from the CA1 of the hippocampus.
There is one additional question here. from Lexi. That is, how long did the training take for the rats to begin expressing measurable cell assemblies? Oh, wait, and then Lavanya also has a follow-up question. For the clustering approach used, how many data points are used?
Okay. Okay, about the first question, how many days would be necessary to be able to detect the cell assemblies? So the one important feature, like when we use a template match, for example, it's very important that critically important that we can make some good templates, meaning that animals should behave somewhat stereotyped behavior. So for this type of experiment, it requires about 1 to 2 weeks of the training. And for this experiment, I'd say maybe 3 to 4 days, at least, days of experiments. For this one, like a trace eye blink conditioning, it appears about 1 week. This experiment was a bit challenging because they actually delivered a stimulus like a 50% of chance. So it was a bit challenging, but I remember they started closing the eyes about 1 week later, they started training. So it's about those period would be necessary to reliably detect the templates. But it is very highly possible that the cell assemblies started showing up in the very early days of the training. And the template match is not a very good method to detect that. And also the method that we are kind of proposing here are probably not very sensitive to that, because if I just go back, because anyway, the cell assembly was defined by, as a group of neurons, which being reactivated, repeatedly. So maybe early days of the experiment, the cell assembly may change over time. They may not repeat it so often. So it's very challenging to detect, but I would expect there are probably quite many variety of cell assemblies exist, but we simply don't have a good methods to detect those yet. The second question was about the data points of the clustering. So let me see. So the question was maybe corresponding to this portion, maybe. So let me see. So for this approach, the clustering used the actually the entire recordings of the recording sessions, which was about 2 hours of recordings. So there were quite many spiking information. It may work also for the shorter period of recordings, maybe having, let's say, 1,000 or 2,000 spikes, but I have to check back. Yeah, sorry.
Those are all of the questions. Oh, wait, Lexi just snuck one in. If you're measuring in the CA1 of the hippocampus, how does the propagation of new neurons skew your data comparison between young and old rats?
Sorry, how the propagation? Or
how does the propagation of new neurons skew your data comparison between young and old rats? Oh, new neurons formant?
The formation of new neurons from the neural stems or other cells.
So, okay, so I may not have understood the question clearly, but so the question was about how the addition of the new neurons to the cell assembly, how this going to skew the analysis, or what's the propagation of the new neurons means, like. So maybe the question was, if the cell assemblies kind of keep changing by maybe addition of the new neurons or maybe removal of some other neurons, how this method works?
Exactly.
Okay. Okay. Yes. So if the cell assembly keeps changing, it's very hard to find the good matches. So in all these methods, we are assuming that certain stationality during the analysis period. But yes, I agree. Yes, the cell assemblies can be quite a dynamic thing, so it maybe keep changing, and especially this may be true for the early days of the experiments. And right now, I don't know the good answer to that. If the cell assembly keep changing, how we can keep track of? I don't really know the good approach for that yet.
Thank you.
So Masami, we have probably about 10, 15 minutes late. I know that we're a little bit behind schedule.
Okay.
We scheduled an hour for this, but I think we're having some great conversations. I just wanted to give you it is 2:10.
Okay. Okay. So yeah, I try to finish up the next section. Try to finish quickly. Okay. So so far I've talked about the data analysis. One focusing on the estimation of the neural interactions. The second was detection of cell assemblies. In the last portion, I'd like to talk a little bit about the modeling and the theories, how the data analysis and modeling can work together to deepen our understanding about information processing. So far we have seen that the HEB synapse is the key for the neuroplasticity. But we also want to ask questions, something like this. How the HEB synapse store the memory patterns stably, or we want to ask questions: is there any limit of the number of the stored patterns? Or maybe we want to ask also the question: how does the the sleep contribute differently from the waking? Doing the data analysis can answer many of the questions, but it's not so easy to really tackle these questions just by doing the data analysis. And here is where we're gonna see the value of the modeling and theory, because they can provide at least some partial answers and deepen our insights into these questions. In the earlier part of today's talk, I also showed that about the information geometry method development. We did computer simulation and analyzed the simulation data and compare the ground truth from the simulation and about the information geometric estimate. So the modeling can also quite useful to develop new methods. Okay, but I'm going to talk about how we can deepen our understanding using the modeling and theory. And this portion, because this is the first talk of this series, I want to kind of touch on the very important studies which was done nearly like 30 years ago. But this was the best example I've known so far about how the modeling theory can deepen our understanding. So the idea I want to start from the study done by the Hopfield. He was a solid-state physicist. In 1982, he actually investigated a very simple recurrent network of the binary neurons. So this was the simple picture how his network looks like. So all the neurons are connected to each other, and here we see the 3 neurons. And he assumed that state of the network is represented by co-activity of neurons. So it's very similar to the cell assembly. So for example, let's say neuron 1 is active, neuron 2 is not active, neuron 3 is active. We see 1, 0, 1. This is the one pattern, and we say this is corresponding to one memory. In another occasion, we may see neuron 1 is 0, not active, neuron 2 is 1, neuron 3 is 1. So we see 0, 1, 1 pattern. This is corresponding to another memory item, for example. So he asks questions that how can we implement these memory patterns to the network of the binary neurons? His answer was something like this. So he actually showed that if we modify the connection weights following the Hebb's postulate, meaning that we modify the connection weights when 2 neurons are co-activated, we can store the memory items, memory patterns, as a local minima of this network, energy surface. So here is the kind of picture how the energy surface would look like. It's actually, there's maybe lagged surface. Then if you look down this surface, it may look something like this. So there are many small, local minima, and there are the area which is attracted to those local minima. So these different local minima would correspond to different memory items. And because when we start from somewhere nearby, it is attracted to this local minima, this is called attractors. In another way, for looking at this energy surface, you can cut this surface. It may look something like this. Okay. So we're gonna see there's some local minima. They are each corresponding to the memory items, something like this. Okay. And when you start from the nearby location, they will be attracted. So therefore, this is called attractor dynamics. But the main part of his contribution is that by combining the Hebb's idea from neuroscience to the energy idea from physics, he actually showed that by changing the connection weights following the Hebb's postulate, we can indeed store the memory item as a local minimum of the energy landscape. So we at least see some mechanisms why Hebb learning works. Okay. For the question about the capacity of the Hopfield network, there are great studies done from the statistical physics, and using the theory of the phase transition, they actually estimated the number of the patterns which can be stored in the Hopfield networks. They actually derived this very famous phase diagram. Here, the x-axis, this is showing the capacity of the network. Capacity alpha is defined by the ratio of the number of the patterns stored over the number of neurons. So for example, let's say we have a network of 100 neurons, and if we can store 10 patterns, the capacity will be 0.1. Okay, so that's what you see here. This axis, this T is the temperature. Basically, this is the measure for the randomness of the network. When the temperature is 0, network behaves completely deterministic. When temperature is high, network behaves quite randomly, stochastically. And they have identified 4 different phases, A, B, C, D. And this is corresponding to the cut of the energy landscape, corresponding to something like this. So what they showed was that when we are in this area A, region A, where temperature is low and we don't try to store very many patterns to the network yet, that's A, energy landscape looks something like this. So we can see all the memories are stored as like a global minima. So that's very good. So this works as a memory network. When we move to the area B, it looks something like this. So the memory item can be still stored, but it's getting quite shallow. Yeah, and we actually see something different is really getting deep. These are the spurious memory states. So those are the memory states we did not implement, but network automatically generates as a false memory. Okay. We go to further into this C. That means we try to put more memory items, but also increasing the randomness of the network. This the attractor disappears, so this became unstable. So we say C does not work anymore as a memory network. And finally, we come to D, where we go to the very high temperature. All these structure disappears, just one single the energy surface. So nothing doesn't work as a memory network. So we can say if we try to put too many memories, there's a phase transition from B to C, which the network forget all the memories at once. And they estimated, especially for the 0 temperature, they estimated maximum capacity about 0.14, meaning that, yeah, if we have 100 neurons, we can put up to about 14 patterns. But when you try to put 15th pattern, network collapse. So that's what they derived. So this was, again, very deepens our understanding of the behavior of the network. Okay, I have just a few more minutes. I just want to briefly touch on this another important work done by the Hinton and Sianowski when they were young. They are very famous now. They actually came up with the idea of the extension of the stochastic Hopfield network, which we kind of already talked about, stochastic area. They actually investigated a slightly different way, meaning that they actually divided the network of neurons into the visible units, the units which we can see, and the hidden units that's here. So we can say that these are the visible units, let's say corresponding to inputs. So maybe we can say these are the visual cortex. This portion is the output neurons. So maybe you can imagine these are the motor neurons. The important part is that they have the interaction with external world directly. Visible units have the interaction with external world. This does not. So this is more like a prefrontal cortex. Okay. So they investigated how the Boltzmann machine store the memories. And interestingly, they actually found that the change of the connection, which try to minimize the energy, go down to the energy, can be summarized by two terms. First term, again, this is corresponding to the Hebb postulate. We have the activity of two neurons. So we take a product. That means we look at the joint, activities. Here we have the positive. So meaning that when two neurons are active, we increase the connection weights. But here we have the negative sign, meaning that when two neurons are co-activated, we're going to decrease the connection weights. And we see that this should happen when these two areas of the visible units are connected to the external world, meaning that when network is awake. Yeah. But then this phase should happen when these visible units disconnected from the external world. So we can say this is sleep. So to summarize, the first term is corresponding to the Hebbian learning during wake phase. Second term is corresponding to the Hebbian unlearning during sleep phase. This actually inspires the Nobel Prize winner, Francis Crick, and at that time, and he proposed that interesting idea about we dream for forgetting. So this is how our sleep looks like over one night. We know that sleep, we go through the different sleep stages. Nowadays, actually, these are clustered together to the sleep stage 3. But anyway, the point is that we go to the non-REM sleep, come back to REM, another non-REM sleep, come back, And the REM sleep is the time that we often experience the vivid dream experience. So from this idea, during sleep, we do the anti-Hebbian learning. This is basically, unlearning. He was kind of proposing that by dreaming, we actually forgetting the things. This might be interesting because as we see, we see there's a lot of spurious memories automatically generated when we implement the memories. By, making the energy surface shallower entirely, these may be erased. So the spurious memory can be erased, but the deep, memories, what we implemented, may still remain. Yeah. So the Hebbian unlearning may erase these spurious memories. Finally, sorry, maybe I'm using up all the times, but I just want to point out that all this idea was based on the physics idea going down the energy surface. That was what we saw before. But interestingly, this energy function is indeed to take the form like this. And as you may aware, this is the form of the KL divergence between the two distributions from the wake and the sleep. So the change of the connection weights is indeed minimizing the KL divergence. And the minimum is achieved when two distribution Q and X becomes the same. Okay. So from the information theoretical point of view, the learning of the proximal machine can be interpreted by minimizing the distance between the two distributions. So we now see another insight from the information theoretic point of view. So I'm kind of speeding up in the end, I'm sorry, but I just want to summarize today's talk. So we said that computational neuroscience investigates the brain function using the mathematical tools and theories. And I have introduced two examples for the data analysis. That was estimation of the neural interactions and detection of cell assemblies. In the very end, I described some important previous work, how the modeling can help deepen our understanding. Example came, as like attractor dynamics ideas, capacitive network, and we talked about wake-sleep algorithms. So we can say all these insights will help understand how our brain works and also advance, the modern technologies, such as something like this. And I know that a few weeks later, my colleague, Dr. Ruchak, I think he will talk more about some interesting study using the deep nets to analyze the animal behaviors. Okay, thank you so much. That's all of my talk today.
Thank you so much, Dr. Tatsuno. Lexi has another question for you. Is the attractor concept related to the electrochemical imbalance that precedes neuron firing? as the voltage potential changes across the soma of the cell.
Okay, so the question was, is this attractor dynamics is related to the gap junction thing, electric? Sorry, could you maybe repeat the question again?
Yeah, basically, I'm just wondering whether that concept of why it's an attractor is related to the change in voltage across an assembly of cells.
Okay, so so we are talking about the very simplified system where neurons activity can be represented by binary states, 1 or 0. So yes, 1 is like active states, active voltage, 0 is the non-active voltage. Okay, then in this framework, yes, we can relate the attractor of the system to the so we can, what we see was that these attractors corresponding to these patterns, which we embed using the Hebbian learning.
Okay, so an attractor would be called a pattern, and then one pattern would have more or less energy than another because fewer or more neurons are firing at the same time.
Okay, yeah. So the so in reality, there will be the like a different depth for each embedded patterns.
Okay.
So yes, if it's like a deeper, or it has a large basin of attraction, that is like a like a stronger attractors, which probably more stable states for the network. So there will be the different properties for the different patterns embedded. Okay. I don't know. Sorry, maybe I may not be answering your question very well, but sorry.
Do we have any other questions? All right. Well, thank you so much, everyone.